give people a minute to filter in. All right, so as people come in, I wanted to say hello, everyone. Good morning, good afternoon, and good evening, wherever you are. Welcome again to the Long-Term Animal Research Seminar Series. We're going to continue the set of seminars from this semester with talks from Dr. Guill McIvor and Dr. Elizabeth Archie. And before I introduce our speakers, I have a few announcements. First, we are evaluating the effectiveness of this series and we need your help. So please, if you can, take 3 to 5 minutes and fill out this survey, which I'm going to put in the chat right now. And we're using this as an evaluation to evaluate our broader impacts, and the results from the survey will help chart our path forward. So please take a minute to fill that out, and if you missed the link in the chat, it's also on our Twitter page where we announce the surveys. Thank you, Matthew. And secondly, if you're participating via Zoom, you should see a Q&A tab at the top or bottom of your screen. If you open that tab, you'll be able to type any questions you have, as well as see and upvote other people's questions. And at the end of the talk, we'll go through those questions, starting with the ones with the most votes. Lastly, recordings of all the talks will be available on YouTube shortly after they conclude. So if you need to leave early or know others who are unable to attend live, this talk will be available for viewing and reviewing after it's complete. All right, now that the announcements are over, I'm excited to introduce our first speaker of the day, Dr. Guill McIvor. Guill received his PhD from the University of St. Andrews, where he did a fascinating project assessing nesting decisions of hooded crows. And in doing so, he worked with power companies to help minimize power disruptions caused by these crows' nests. He then held research positions at the University of Exeter and Newcastle before starting his current position as a postdoctoral research fellow in the Wild Cognition Research Group at the University of Exeter. Guill has pursued a variety of different research questions in corvids and other birds, including addressing how social environment affects learning about risks, how individuals choose nest sites, and the role of group composition in the movement of flocks. Guill also helped create the Cornish Jackdaw project, where he works to investigate and better understand cognitive evolution in the animal kingdom. So I'm really excited to hear more about these projects today. So Guill, go ahead and take it away when you're ready.
Thank you very much. Are you seeing the correct screen just now, Liz?
Yes.
Marvelous. Okay. So thanks very much for having me along today. I'm going to talk to you about a study that I've done of eggs in the jackdaws. Although I've just lost my presenter's review. This is an inauspicious start. Please excuse me. We'll try again. There we go. And we're off. Okay, so first of all, I'd like to start with some acknowledgements. Firstly, I'd like to...
You're not sharing screen with us. We can't see your screen right now, sorry.
My bad, my bad. I tried to fix the problem, but I forgot about the screen sharing. Let's try again. How about now?
Perfect.
Have I got the right screen this time? Hooray. Thank you for bearing with me. Okay, so first of all, I'd like to thank our group leader, Alex, who has employed me for all these years and allowed me to do the job that I love, and also for our funders who've supported us during my time doing this research. The project I'm going to talk to you about today, my collaborators have been Julian Trushanko, who's a sensory ecologist who's based in our department here in Exeter, and also Madeline, Max, and Nafi, who worked on this study as part of their undergraduate projects. Finally, I'd like to thank the PhD students in our group who've all played a really big role in getting the project to where it is today. To give you a brief overview of our system, I started off with the Cornish Jackdaw project back in 2012, when I joined Alex's team to help in his fellowship looking at the evolution of intelligence in the crow family. Jackdaws made a really great study species to do this with because they were abundant in our local area. They're highly social and they'll readily use nest boxes like you can see in the far right of the screen. And from there, our projects expanded and we now focus on a wide range of topics like you can see here. But while my main work at the moment is looking at the impact of social relationships on the collective behavior of the birds, today I'm going to talk to you about a little study I've done looking at the eggs of the birds. So why eggs? Well, eggs have fascinated people for centuries. And when you look at a picture like this, it's really easy to understand why. The ancestral egg is thought to have been white. And from there, it's evolved into a diverse array of colors and patterns. And the use of pigments in this is believed to be linked to things such as crypsis, recognition of the eggs when there's brood parasitism. And more recently, people have started to wonder if it could be to do with signaling. Now, this last one has really piqued my interest because when I looked at a lot of jackdaw eggs over the first few years of the project, and I came to notice that they had a huge amount of variation in their size, shape, and the color of the eggs, and also the maintenance of the color. So you have some eggs, such as in the top right here, which are really washed out. But then you have others, such as the one in my hand, which are absolutely gorgeous in the intensity of the color. And so I was interested to see what was influencing this variation and what consequences there might be. And this is where it pays to have clever friends such as Julian, because he had realized and published a paper in 2014 where he discovered that you could use the photograph to calculate the volume of an egg. And not only that, as well as looking at the volume of the egg from the photograph, you could take advantage of the image analysis tools he had developed to look at the color. And you could apply something called adaptive thresholding that allows you to separate the background color of the egg from that of the maculation. Maculation is the name that we give to the spots on the egg. And so you could look at these two colors separately as well. And remarkably, in these eggs that you see in the picture, all of the diversity in color that we have comes from just two pigments. So biliverdin is responsible for the blues and the greens, but protoporphyrin is what gives us the reds and the browns. Interestingly, biliverdin and protoporphyrin, they seem to have good cop, bad cop rules in the physiology of the birds. So biliverdin is an antioxidant, but by contrast, protoporphyrin is a pro-oxidant. And in physiological terms, it's probably having too much or too little of something is a bad thing. But as a general rule, it's likely to be that having more biliverdin is beneficial to birds, whereas having too much protoporphyrin is a bad thing, although they may need some if they want to mark their eggs with spots for camouflage, etc. And because biliverdin is a finite resource and therefore costly to allocate to eggs, a theory developed about 15 years ago called the sexually selected egg hypothesis. And this was built around the premise that a female might use her eggs as a signal of her, or the coloration of her eggs as a signal of her investment in the clutch to a partner, and therefore encourage a high level of investment in the reproductive attempt from him as well. And they did some correlative and cross-fostering experiments with pied flycatchers, and they found some evidence to support this. Manipulation of the plumage badge on the males would encourage the females to lay a more heavily pigmented egg, whereas if you put a super blue egg in the clutch, then the females would also provision more later on. And they have found evidence also that these egg colors link to the maternal oxidative state and the elements that she's putting into the egg as well. And further evidence has come from studies from species such as the spotless starling and also blue-footed boobies. But it's important to add that plenty of people have looked at this and they've found no such evidence. They've also expanded to look at whether or not these same hypotheses and reflections of condition might exist with protoporphyrin depositions in other species, and that has an even less clear picture. Nevertheless, I was intrigued and I wanted to investigate how the coloration of our jackdaw eggs might vary in relation to maternal condition and quality, and whether or not there was any impact of the color of these eggs on the subsequent growth and survival of the chicks. I predicted that the females in better condition would be laying bluer eggs, while chicks that hatched from bluer eggs would be more likely to fledge and would grow to a larger size. Our system gives us a really great avenue to look at this because the females are long-lived and they come back year after year. And I'm going to present data from 3 years today. We monitor our nesting attempts really closely. So every day I check the nest for eggs, and when the eggs are laid, I climb up with a Sharpie and number them. And then 8 days after the clutch was initiated, I take the entire clutch down and I'd photograph them with a scale bar and a color standard. And the camera that I'd used is one that had been calibrated by Julian, so that the colors that we analyze are mapped to the visual field of the Jackdaw, and we consider them in a way that is representative of how the birds are perceiving them. And I calculated the opponent channels based on PCA of the wavelengths that are being reflected to generate the color measures that I'm going to report today, like the blueness and the redness. And after the eggs hatch, we monitor the growth of the chicks and mark them so we can track each individual and have a clear link from the egg to the arising chick as well. So we weigh them every 3 days from the day they hatch until they reach day 26, when they can be either magnificent like this chap here or distinctly underwhelming like this boy. And in between hatching and fledging, we also go out and spend numerous evenings creeping around the countryside. This is me with a team of helpers, and they're using sponges on poles, very low-tech approach to trap the females in the nest box, so I can take them down and get their biometrics and mark them if they're unmarked. Now, jackdaws are exceptionally synchronous species when it comes to breeding, and almost all of their eggs are laid within a 14-day window in April, with some stragglers that come in at the end. And we might expect the highest quality birds to be at the front of the pack and the lower quality ones to be at the end. So I started my analysis by looking at the general features of the eggs and how these features changed over the course of the laying period. And in all 4 graphs that I'll show you here, the laying date is on the x-axis. What we found was that the background blueness of the egg decreased over time. But by contrast, the redness of the spots would increase the later the laying date was, while the proportion of the surface of the egg that was maculated would decline slightly over the laying period, and there was a very, very slight tendency for the egg volume to decrease also. But as jackdaws are a species that has asynchronous hatching and brood reduction, we also expect them to be probably favoring the earlier eggs in their broods over the later ones in the allocation of resources. So if we change the x-axis so that instead of the lay date, we have the lay order of the eggs from 1 to 6, you see that we do indeed find that there is a strong impact of the lay order on pretty much all traits. So there's a decrease in the background blueness over time, while the redness of the spots increases. And the strongest traits that we see is that the proportion of the egg that's maculated decreases later in the laying order. And after the second egg, there's a big drop off in the volume of the egg also. So based on these last 2 slides, it's clear that collinearity exists in the features of the eggs, the date on which the egg was laid, and its position in the laying sequence. So to get at the heart of whether or not the egg features were changing over time, I considered only the first leg egg in a final analysis. And when I looked at this, I found that there was no decrease actually of the level of spot coverage or egg volume over time. But the females that were laying their eggs or initiating their clutches later on were laying less blue eggs and eggs that had redder spots than the ones that were initiating their clutches earlier. Therefore, it does appear that egg color may be linked to maternal quality in some way. And the link between the blueness of the background of the egg and the redness of the spots is really, really tight. This is probably the tightest relationship we're ever going to identify in jackdaws ever, I think. And so because they seem to be so tightly linked, I'm only going to look at blueness going forward, because this is what interested me most. And one last thing in this analysis of all the eggs is that we found that the eggs from the later clutches were less consistently blue. The graph I have here is an interaction plot, and you can see the broken light blue line at the top. That is representative of the females that were in the earlier part of the group, or above average, or earlier than average in their lay date, whereas the solid line is those that were later than average in the date of clutch initiation. So it does appear that females that lay their clutches later find it harder to sustain the blueness of the clutch, therefore potentially indicating that, yes, it is a limited resource and could be linked to female condition and quality. But what of the maternal traits? So I started building models that use a subset of the nesting attempts where the female was captured, and I'd always factor the number of days that had elapsed between clutch initiation and the date that the female was captured in my analysis. So the females, they generally make very heavy weather of raising a family. They lay usually 4 to 6 eggs, but typically most pairs will only fledge 2 or 3 young. And in doing that, they lose about 10 to 20% of their body weight. So I always factored in how many days had elapsed when I was considering their body condition. And what I found was that there was no influence of condition whatsoever on the blueness of the eggs. If anything, there was a very slight decrease once you factored in days since clutch initiation. And that's perhaps unsurprising, because if blue eggs, and blue is best, and you expect the females that lay bluer eggs to be rearing more chicks, then they're probably going to have to be working a lot harder to revision their brood, and therefore losing condition a lot faster. And we see some support for this in this graph, where you can see that most of the birds in a population will fledge 2 to 3 chicks. But there's a real step in the level of blueness that you can see in clutches when you compare the ones that fledge 2 chicks to the ones that fledge 3. But there are also other factors that influence the blueness of the eggs. And we found that the larger females laid bluer eggs. That's just a pure structural size of weight, tarsus, and wing. And likewise, the eggs from larger clutches were bluer also. And as we saw before, clutches that were initiated earlier in the season were bluer than those that were initiated later. So in summary, it does look like we have evidence that egg blueness is linked to common measures of maternal quality, be that clutch initiation date, body size, and clutch size. And that was really encouraging, but what would really help support the idea that bluer was better is if these traits linked to the growth rate and survival of the chicks. And it was Madeline who had a great idea about how we should address this, because she noted that for the first and the second hatched egg, the survival rate was pretty much 80%, whereas the 4th, 5th, and 6th laid eggs pretty much had sub 10% survival. But the 3rd laid egg had 50-50 chance of surviving. So when we looked at the 3rd laid eggs, we thought that would give us the best opportunity to see if having a bluer egg increased your chances of survival. And what we found was that it really didn't. There was absolutely no influence whatsoever of blueness on the probability of survival, nor was there any influence of the volume of egg on the probability that the chick would survive. The only thing that we did find was that early clutch initiation date was likely to improve the survival probability of the 3rd hatched chick, or the 3rd laid egg. And even if I expanded this analysis out so that we looked at all of the eggs in the brood, the results stayed consistent. But what about other effects? Might the chicks at least grow better? Well, sadly, that returned rather underwhelming results as well. Chicks from bluer eggs grew to no larger size than ones from less blue eggs. And blueness was such a terrible predictor in all of these analyses that it never really explained much more than 1 or 2% in the variation of survival or growth. We did find, however, that chicks from the larger eggs grew to a larger size, as well as additional influences of far less interesting predictors. So the ones that hatched earlier grew larger, males grew larger than females, and we did find a colony effect as well. So our more urban colony did much worse than our more rural ones. So to sum up, the egg blueness does appear to be linked to maternal quality in these birds, but despite this, egg blueness had no apparent impact on the survival or growth rate of the arising chick. Future directions I'd like to take the research in are to link the blueness of the egg to the maternal and egg oxidative state, as well as looking to see if it may be reflective of the yolk contents and things like immunoglobulins that are being added, because that's currently a big gaping hole in the data set. We also might run future undergraduate projects looking at any links between plumage traits and the egg color. And hopefully with one more year of data, I'll be able to start looking at the influence of senescence on the egg color traits as well. And that's something that I think would be really great to get at, because I don't think anyone's ever done that before. So, thank you very much.
Great. Thanks so much, Guill. We have some questions coming in. First, there is an anonymous question that says, is it possible that the sexually selective blue egg hypothesis is simply wrong?
Yes, very much so. I think that there has definitely been a tendency for the people who want to see it most to be more likely to identify it in the data sets. And a lot of the like most strongly touted examples and the foundational examples came from very small sample sizes as well. So it seems like the most robust studies are the ones that generally find that there is no effect. But nevertheless, it does seem to be that there is some tie to female quality. So it's like it could be a signal that the birds attend to, but I just think that the evidence that we have today suggests that there is no attendance to it by the birds, which is a bit odd because we can see the differences and the birds can probably see the differences, but they don't seem to act upon them.
Gotcha. Okay, so we have another question from Mark Hauber, who says, there are several colorful non-avian dinosaur eggs that predate bird evolution. What do you think this means for the ancestral or baseline avian egg coloration?
I had no idea about that. This suggests that we might need to rethink things a bit. Yeah, I wonder what we could find out about, like, what the method of parental care was for those eggs as well. Because I think the general assumption is that the dinosaur eggs maybe didn't need to be as camouflaged because they were often just being covered, and so there wasn't so much selection pressure for those things. So that's rather turned things on its head for me.
Yeah, I think that's really interesting.
I can't come up with a good answer. Yeah, that's
that
great.
Is all right. Okay, one last question from Katie Fowler, who asks, is there any diet change over the breeding season that could be linked to the egg color change you see?
Yes, so the parents, like, to begin with, I mean, they're all laying in such a tight period of time, so we think that the chances are they're probably all foraging on similar things. Although we did see, if I hop on to this last bonus slide, when I looked at the spot coverage proportion, there seemed to be an effect of the site on the proportion that the eggs were maculated. And so it seems like the cleanest living birds over at this site here have less spotty eggs or lower spot coverage of eggs than the ones that are on the farm where they're grubbing around in barns and on campus where they're spending a lot of time on concrete. So there are likely to be dietary effects. And it'd be interesting to see, we have RFID tags on these birds as well. So we can potentially limit their access to supplementary food resources in different groups, so we can allocate access to supplementary food that could help with antioxidant levels and the ability to allocate biliverdin eggs. And so, yeah, we could start to try and manipulate that experimentally and see if it is linked to what the birds are eating.
Yeah, that'd be really cool. All right, so thank you so much. I think we're going to stop there for time. And I will introduce our next speaker. So I'm really pleased to introduce our next speaker, Dr. Elizabeth Archie. Beth is a professor in the Department of Biological Sciences at the University of Notre Dame, and she's a project leader of the Amboseli Baboon Research Project. Her research focuses on the evolution of social behavior by integrating long-term data with immunology and genetic tools. And currently her group is focused on understanding how social organization affects the spread of parasites and microbes within and between groups, as well as how social relationships affect individual health and survival. And what I really appreciate about Beth is how she methodically approaches questions and uses these integrative approaches to answer big questions in biology. So I'm thrilled to welcome Beth today to hear more about microbiomes and survival in wild baboons. So Beth, take it away.
Great, thank you, Liz. That's really nice. And I want to thank you guys for inviting me. It's a real honor to be able to speak to this crowd. And I've really enjoyed watching these seminar series myself, so it was a great idea to put this together. All right, let me share screen. And does that look right to everybody? You seeing my slides? Great, all right. So I want to start by thanking the members of my lab. I've had the chance to collaborate with a lot of great people over the years, but in particular, of course, the people pictured here have contributed to what I'm going to talk about in very large ways, sometimes in small ways, but all of them have contributed. And I feel really lucky to be able to work with such a smart and fun group of people. So just wanted to give a little shout out to them. So working on this theme of long-term data, you know, I'm preaching to the choir here, but in the 1960s, early 1970s, there was a sea change in population biology where for the first time, people began to look at the lives of individual animals and study them in an unbiased, concerted way and follow these lives longitudinally. And this allowed us to see things in ecology and evolution that we'd never seen before, such as measuring lifetime reproductive success in long-lived animals, actually looking at life history trajectories in individuals, or looking at how traits transmit from one generation to the next. This ability to sort of watch animals longitudinally also has parallels in human cohort studies, especially human cohort studies to understand social and environmental drivers of health. And as a result, a number of these long-term animal studies also draw conclusions that can be relevant for understanding human health and well-being. So I'm very lucky to help lead one such project, which is the Amboseli Baboon Research Project, which was founded in 1971 to understand long-term changes in primate social behavior, ecology, demography, and genetics. This project is located in the Amboseli ecosystem, which is in southern Kenya. And it's led by a team of scientists, Susan Alberts at Duke, Jean Altman at Princeton, myself at Notre Dame, and Jenny Tung at Duke. And our data are also collected by a team of observers. Everybody pictured here contributes to data collection in different ways, some by driving observers, some by cooking, and some by actually collecting observational data and behavior and biological samples. And in particular, I want to acknowledge 4 people, Raphael Mututua, Kinua Warotere, Longbida Siodi, and Sarah Siolel, who together have contributed the vast majority of behavioral ecological data I'll show, and also collected a lot of the biological samples. So what this team does is they're out there 52 weeks a year, 6 days a week. We do give them some time off, but they're out there collecting data, trying to understand what's going on in the lives of animals from the day they're born to the day they die. And they're collecting data on demography, on individual reproduction, on social relationships, on reproductive patterns, on group ranging patterns. You name it, we try to collect it. And since the onset of the study in the early 1970s, we've collected these kinds of data on about 2,000 different animals. And at any given time, we collect this information on roughly 300 animals currently living in 5 different social groups. So in case you're not very familiar with savanna baboons and their society, is just, here's a quick primer. So baboons live in social groups that can range quite a bit in size, from as small as 20 animals to up to 150 animals. Those groups consist of multiple adult females and males and their offspring. And females are matrilocal, so they live together with their mothers and their sisters and maternal kin in a stable social group. Whereas males, when they reach sexual maturity, they disperse to a new group and often may disperse repeatedly across adulthood. So today, what I want to do is talk to you about some projects we have been tackling under 2 sort of big questions. And these are projects that both really heavily leverage these long-term data and the long-term perspective we get from the Amboseli Baboon Project, and are projects that are sort of coming to fruition or have come to fruition in the last year. And so the first projects I'll talk about are trying to understand the dominant drivers of gut microbiome dynamics in the Amboseli baboons. And then second, we'll sort of turn our attention away from the gut microbiome to look at developmental and behavioral consequences arising from early life adversity. All right, so diving into the gut microbiome. I'm sure many people in the audience know that gut microbiomes are highly diverse and dynamic ecosystems. And from these dynamics emerge a series of life-sustaining services for hosts, including digesting food, the ability to produce certain key vitamins, pathogen resistance, the ability to actually combat toxins in some foods. So the list of how the gut microbiome contributes to health is growing every day. It's also really true that these communities are very diverse. So for instance, in the baboon samples that we have, we are able to detect members of 30 different bacterial phyla in our samples, across all the samples. Moreover, these bacterial communities are quite dense. It's one of the densest microbial communities on Earth, and the cells have a wide range of interactions with each other. Some microbes compete with each other for the same resources. Other microbes collaborate to such an extent that they can't even be grown in isolation. And one recent paper by Rainey and Quistad said it best, I think, where they said that attempts to comprehend the multiplicity of effects, including those on the patterning of diversity in the microbiome, addle the mind. And it's true. A lot of people, when you try to first understand what's going on in microbiomes, your mind feels addled. And I think one reason for that is that the types of data we have had have made it hard to see the forest for the trees. And in particular, most microbiome data sets are cross-sectional that consist of a single sample from a single, or from animals in a population or a set of populations. And this snapshot perspective makes it hard to understand sort of the general organizing principles. It's a bit like if you dropped in on a rainforest or a coral reef for just one day and collected data and never revisited, it'd be really hard to understand what's going on. So this is a place where long-term studies can really contribute by contributing longitudinal data that help reveal the generalized and organizing principles that shape microbiome dynamics. So just as, when we're trying to understand forest change, as you can see in these landscape history diagrams from Harvard Forest, and how they're shaped by their environments and by anthropogenic factors, we need similar longitudinal data on the microbiome. So this is something that my lab has been working to gain together with some collaborators. This was definitely a huge team effort. We've been gaining time series data on the gut microbiome, and in particular, the data set we're working from consists of over 17,000 fecal samples collected from just over 600 baboons over a 14-year span. And these plots on the right show you a subset of these time series. So a dot here represents a given sample, either from a juvenile or from an adult, as a function of baboon age. And on the y-axis, we have each unique individual in the data set. I particularly want to call out my PhD student, Mona Dasari, who did all of the DNA extractions for these samples, which was a Herculean task. And once the DNA were extracted, we performed 16S ribosomal RNA gene sequencing on them, ran them through the data2 pipeline. For microbiome people in the audience, we ended up with about 40,000 sequencing reads per sample to characterize their composition. So there's a lot of questions that can be answered with this data set. And in fact, we are tackling a bunch of different questions on these data. But one that I'll tell you about today is, simply put, does everybody's microbiome seem to work the same way? And this is work led by Johannes Bjork, who's been a postdoc in my lab. So the basic idea here is that if you have a set of hosts living together in the same population, experiencing very similar environments, similar perturbations, do their microbiomes change similarly or change in synchrony? And if they do, that's important because it tells us that microbiome dynamics should be fairly predictable across hosts in the same population. And if you design a microbiome therapy, it's likely to work the same way in different hosts. So answering this question, again, requires that we have longitudinal data from animals in the same environment. And indeed, that is true for this data set. So the samples that are in this large data set come from subjects that lived in 10 different social groups. But those social groups had highly overlapping home ranges over the course of the study, as you can see by these 95% kernel density maps of the home ranges of these groups over the time period. And moreover, the members of each group also have highly similar environments. And that's because the members of a baboon group tend to travel together across the landscape, encountering similar resources at the same time, and exhibiting sort of highly coordinated behaviors. All right, so these are some forces that might lead to microbial synchrony. There's also, however, reasons to think that each person's each baboon's microbiome might be highly personalized or idiosyncratic, okay? And there's a couple reasons to think this. One reason is that microbes have this awesome ability to swap genes with each other and gain new functions. And as a result, a microbe with a given 16S sequence in one host might not actually fulfill the same ecological role across hosts because of this flexibility. Just as important, or probably just as important, hosts themselves have the ability to curate and shape their microbiomes through their genomes and through their immune systems. And this host control and the uniqueness of individual hosts may lead to considerable microbiome personalization. If that's what we see, this makes it a lot more challenging to predict microbiome dynamics, because it means those dynamics might be dissimilar across hosts. And it also poses challenges for designing microbiome therapies. We might need personalized approaches. For instance, if you were going to design a microbiome like a probiotic. Okay, so we set out to tackle this question on the data set, and I'll show you what we saw. So first, we do see evidence for some modest microbiome synchrony, especially across annual seasonal cycles. So in Ambicelli, the habitat is shaped by predictable seasonality. We have a 5-month dry season during which it doesn't rain, followed by a 8-month, actually I can't add anymore, a 7-month long dry wet season that's highly unpredictable. Where sometimes there's a lot of rain and sometimes there's not. And these are 2 photos that I took from the end of our driveway at camp that just sort of shows you some of these seasonal extremes. So together with this seasonal differences, we have a predictable shift in the foods that are available to baboons and the diet that they eat. So this is a graph of the abundance of common baboon foods across the 14 years of the data set. And the take-home message is that during the dry seasons, baboons are relying on a predominance of underground growing grass corms. And during the wet seasons, you see these orange blobs, and those are representing a shift to eating new growing grass leaves. So we see this sort of predictable major shift in diet. So if we take all 17,000 microbiome samples and throw them into a giant PCA, what we see is some separation by season along this first principal component. And so in particular, we see samples separating off to the left-hand side of this principal component during the dry season, whereas during the highly variable wet season, they're sort of distributed along the entire principal component. So if we take this principal component and plot it over time, we see the emergence of predictable seasonal cycles. So on this plot, the x-axis is the 14 years of the data set. The y-axis is that first principal component of the microbiome. And the orange line is a smooth fit to those seasonal shifts. In the background, you've got the brown and green bars, which show seasonal shifts from the dry to the wet season. And the blue bars show rainfall during the wet season. So because these cyclic changes are nonlinear, we need to model them using generalized additive models. And when we do that, we find that seasonal differences explain, about 7% of the deviance in that first principal component. If we look at other traits of the microbiome beyond that first principal component, for instance, the abundances of the most common phyla and families, seasonal shifts are explaining about 2 to 6% of the deviance. So we're explaining something in the data, but we're certainly not explaining everything. And that is, I think, in part because we do see considerable idiosyncrasy or personalization in microbiome dynamics, despite the fact that these hosts are all living in the same place. So one way to see this is through an autocorrelation analysis that compares microbiome similarity as a function of time between samples. And so I'll sort of step you through this plot and then show you the data. So on the y-axis, we've got a measure of community similarity. More similar communities will appear at the top of the y-axis as a function of months between when two samples were collected. So for instance, you can have samples collected in exactly the same month and year over here at 0, or samples collected 108 months apart or 9 years apart. So if I plot our data on here, first I'll show you what we see for samples collected from different baboons living in different social groups. And this sort of gives us like a baseline level of similarity in the population. And what you can see is that indeed, for these samples from different baboons, they are most similar to each other when collected in the same month and year. And that similarity declines somewhat as you go 1 month out, 2 months out, 3 months, 4 months, and so on. And then you see these sort of subtle peaks at 12-month intervals that reflect that seasonal similarity. And it shows you that basically a sample from the population is similar to itself in that month, and then similar to itself again, 12 months later, 24 months later, 36 months later, and so on. I want to contrast this with samples collected now from the same baboon. So these are samples collected from the same individual. And what I want to really draw your attention to is these points up here. These are samples collected from the same individual in the same month and year, which are substantially more similar to each other than are samples collected from different individuals over the same time period. This individual signature does fade quite quickly, but it always sort of remains stronger than differences, similarity between samples from different baboons, even 2 or even 3 years apart. This individual signature is maintained. And that suggests there's considerable personalized dynamics in the microbiome. You may be wondering about social groups. I always am, and I'll throw these on here. So as a reminder, members of the same social group are experiencing similar environments as they travel through the landscape together, encountering similar resources and eating similar foods. And we do see that members of the same group, so now we have different baboons in the same group, they do have more similar microbiomes compared to baboons from different groups in the orange lines, but not nearly as similar as samples from the same individual. So it would be nice to sort of put some numbers on this, like how important are population level versus individual level traits. And one way we've tried to do that is by building hierarchical GAMs that allow us to sort of look at the relative effects of population, group, and host level factors. And so what I'm going to do is show you the deviance that we explain if we first only control for population level factors such as climate, rainfall, seasonality. Then if we add population and group, and then finally population and group and host, and look at how much deviance we're explaining. I'll do that for the first 3 principal components of the microbiome, as well as the 5 most common phyla. And what we can see is that at the population level, if we're only looking at these population factors, we're explaining less than 10% of the deviance in the data. However, once we start adding individual host, we're often explaining substantially more deviance, sometimes more than 50% of the deviance in the data. If you're wondering what deviance is, it's a measure from GAMs that's basically parallel to R². So what these data show us so far is that even when hosts are sharing the same environment and resources, gut microbial synchrony is relatively modest. And in fact, each baboon's microbiome seems to be changing in its own personalized way. We would anticipate that this personalization should be even more extreme for humans, since we're all living in different houses, I mean, especially now, and eating different foods and those kinds of things. And so the fact that we're basically seeing this personalization, even in a system where the environment is relatively shared, is somewhat surprising and points towards the importance of personalization when designing microbiome therapies or when trying to predict these microbiome community dynamics. So what are some of the factors that are then contributing to these idiosyncratic dynamics? I already mentioned horizontal gene transfer, which leads to this sort of ecological flexibility of an individual microbe. And then I also discussed host control, and I'd like to pick up on that a little bit. In particular, talk about another project we've been working on. This is led by Laura Greeneisen, who is a PhD student with me and is now doing a postdoc with Ron Bleckman. And she's been looking at trying to understand the degree to which host genotype contributes to microbiome variation. And in particular, she's been measuring the heritability of microbiome traits, such as the abundances of individual microbes or their presences and absences. So as a reminder, heritability is the percent of variation in a phenotypic trait, such as those microbial abundances, in a population that's due to genetic variation between individuals in that population. So what we know so far about this question is that comes from largely from human studies. So it's measured most often in human populations. And what these studies have taught us is that heritable microbiome taxa seem to be relatively rare. So we only find that about 5 to 13% of taxa in the human microbiome exhibit significant heritability. And the heritability estimates on average tend to be relatively low. So in some studies, the average is about, we're explaining 2% of the variance in these traits using genetic variation between individual people. But there's a couple of challenges with measuring microbiome heritability in humans. One is that, as I mentioned, these phenotypes tend to be measured cross-sectionally in a single sample. And we already know that microbiomes are really dynamic. And so a single sample may not give a great reflection of that phenotype. In addition, it's pretty common to measure heritability in twin studies, which are extremely powerful, but they may have difficulty overcoming confounds between genes and environment because twins may share behaviors and may in some cases share the built environment. So in Amboseli, what we've been doing is using multi-generational pedigrees to help estimate the heritability of microbiome features. And working in Amboseli allows us to get around some of the genetic and environmental correlations in a couple of ways. First of all, members of the same social group have highly shared environments, but actually social groups also contain a very wide range of relatives, some close relatives such as parent-offspring pairs, but also many non-relatives. And in fact, average relatedness in a baboon group is it's around 0.05 or 0.06, so it's pretty low. In addition, we have data on their environments, such as the foods they were eating, what the weather was, and so we can correct for some of these covariates in our modeling. So what we're finding, actually surprisingly, and in contrast to the human studies, is that heritable taxa appear to be quite common. And in fact, we find that 97% of gut microbial abundances are in fact heritable in the Amboseli baboons. This is also true if we look at presence and absence of a given microbe. The heritability estimates we observe are typically low. So for instance, the average heritability of a microbial abundance is about 6%. But the fact that so many taxa are significantly heritable indicates that genetic effects on the microbiome may be more pervasive than we think. I'll draw your attention to just a couple of the traits that were highly heritable. This lists the top 40 most heritable phenotypes. One of them is that first principal component that I talked about in the first part of the talk that exhibits the seasonal fluctuations. There's also a number of taxa in here that are also heritable in humans, such as Christensellaceae, which is down here. Prevotella is among one of the most heritable taxa. So anyway, that's a brief overview of some taxa that were heritable. To summarize this section on microbiomes, what we find is that microbiome dynamics appear to be idiosyncratic to individual hosts, even when those hosts are living in the same environment. Those idiosyncrasies are probably explained by a bunch of things, but in part, they may be explained by strong host genetic effects on microbiome composition and change. And these host level idiosyncrasies may pose challenges for predicting microbiome community changes and for designing microbiome therapies that are going to work well across a wide variety of individuals. Okay, so with that, I'd like to switch gears relatively substantially, move away from the microbes living in the gut of a baboon and switch to the baboons themselves and their lives and their behaviors. And in particular, I want to talk about some projects that we've been working on looking at developmental and behavioral consequences of early life adversity. And early life adversity is a theme that has shaped work not only in my lab, but in Susan Alberts' lab and in Jenny Tong's lab. All of us have been interested in these questions. And that's in part because of a discovery that we made in 2016, which I'll actually tell you about in a minute. I have to give you a little more setup first. So first of all, early life adversity can have profound consequences on how animals live their lives, especially across adulthood, and has major effects on human health. Unfortunately, the experience of harsh conditions, famine, war, abuse, is common in humans and probably contributes to global health and disease burdens. We see the strongest evidence in humans for links between early life adversity and things like cardiovascular disease and schizophrenia. And this phenomenon in general is known as the long arm of early life. As I suggested, this phenomenon is not unique to people. So this is also seen in animals. Long-term studies of animals have been very useful in trying to understand the universality of these trends. And I'll show you one example that we've seen in our own population, which is a very strong relationship between the experience of early adversity and survival. And so to look at this question, we looked at 6 different sources of adversity that could occur in the first 4 years of the life of a female baboon. We looked at aspects of adversity linked to resources, such as the experience of drought in the first year of life, or growing up in an especially large social group where there might be competition for resources. We looked at aspects of family structure, such as maternal loss or the presence of a competing sibling that might divert maternal attention. And then we also looked at aspects of the mom's social environment, such as having a low-ranking mother or a mother who is especially socially isolated. We measured these traits in 189 females in the first 4 years of their life. And then we started the clock at age 4, which is around the time females achieve menarche, and looked at how many remained alive across adulthood, conducted a survival analysis. So what we saw is that for the lucky baboons that had none of those 6 sources of adversity early in their life, their median lifespan was about 22 years. In contrast, for baboons that had 3 or more of those sources of adversity happening in the first 4 years of life, their median lifespan was only 9 years. So this is a more than 10-year difference in life expectancy and was very striking. Ourselves, we were surprised when we saw how strong this effect was. If I fill in what we see for individuals who experienced 1 or more or 2, or 1 source of adversity or 2 sources of adversity, we see the effects on mortality increasing in a stepwise fashion, suggesting that it's the accumulation of these adverse circumstances that is particularly toxic to adult survival. So since this discovery, we've been exploring this in many ways. And as I said, not only in my lab, but also in Susan Alberts and Jenny Tong's lab. And we've been working to identify, in particular, adaptive behavioral or developmental responses that may arise from early life adversity. And I'll tell you about a couple of them that we've been working on in my lab. One is work done by Chelsea Weibel, who's a PhD student in my lab, who asked the question, do females who die young also tend to live fast? Okay, so the logic here is that if a female baboon experiences early life adversity, she may be able to anticipate a short lifespan, predict dying young. And if that's true, it may pay her to speed up her reproduction. In other words, live fast. So maybe reach menarche early, maybe reach first reproduction early, have offspring quickly in order to squeeze in as much reproduction as she can before she dies, okay? So this hypothesis has been around a long time. It's been especially common in evolutionary medicine, in anthropology, and psychology. The hypothesis, has sort of some different theoretical concepts underlying them, but all of them are sort of proposing a link between early life adversity and the timing and pace of reproduction. And that's the way these hypotheses are most commonly tested, is by looking for a link between early adversity and unusually fast reproductive timing, usually early menarche in girls. However, from what we could find, no study had actually tested if that reproductive acceleration was adaptive. In other words, does it lead to higher lifetime reproductive success if you do start reproducing early or fast? And moreover, we hadn't found anybody who looked whether that accelerated reproduction was, and its fitness benefits were truly unique to those who experienced early life adversity. If those fitness benefits of speeding up reproduction are not unique, that just means speeding up reproduction is globally adaptive. So we need to see some sign that there's some unique fitness benefits arising from the experience of early life adversity. So Chelsea set out to tackle this problem, and she did so by addressing sort of 3 key questions. First, she looked, is early adversity actually linked to faster reproduction in the Amboseli baboons? Is faster reproduction actually linked to higher lifetime reproductive success? And then finally, she tackled whether the adaptive benefits of accelerated reproduction are unique to individuals who experienced early life adversity. So here's what she found. So first of all, she looked at a couple of measures of reproductive timing and pace. So she looked at whether early life adversity led to earlier ages at first birth. And then she also looked at whether they led to fast sort of time between surviving offspring or interbirth intervals. This picture is here to remind me to tell you that baboons have 1 offspring at a time. So they invest in 1 offspring sequentially, wean it, and then start investing in the next one. Okay, so what Chelsea found is in fact that there was no relationship between the experience of early life adversity and the timing and pace of reproduction. So you can see here on the x-axis, we've got rising early adversity, and we really see no difference in the age at first birth or the pace of interbirth intervals for females as a function of their early life experiences. However, there is a lot of variation within each of these categories. So there's a lot of variation in that timing and pace. So she next asked whether that variation actually leads to substantial fitness benefits. As we would expect, for a long-lived organism that produces 1 offspring at a time, we found that lifespan explained the vast majority of the variation in lifetime reproductive success. However, reproductive traits, age at first birth and interbirth intervals, did explain a significant amount, an additional 3 to 5% of the variation in an individual's lifetime reproduction. You can see this in these plots that Chelsea's made, where lifespan is on the x-axis and lifetime reproductive success is on the y. You can see that nice linear relationship between how long you live, determining how many babies you produce. And then she split them into individuals who had an age at first birth that was above or below the median. So above the median is blue. And then same with interbirth intervals above and below the median. And you can see that, yeah, fast reproduction does help a little bit in improving your lifetime reproductive success. Okay, so the last question that we needed to know to test this hypothesis is whether the benefits of fast reproduction are specific to females that experience early life adversity. So testing this idea involves looking for a fitness crossover or basically an interaction effect between the experience of early adversity and the timing and pace of reproduction. So there's some key tests here. In particular, we need to see whether individuals who experience early adversity and accelerate their reproduction have higher fitness than those who don't. So that's one thing we need to see. And we need to see that this pattern is different for animals who did not experience early life adversity. If it's not different down here, that just means that speeding up reproduction is globally beneficial. But if we do see an interaction effect, that's evidence that there's a specific benefit for speeding up reproduction under the experience of early life adversity. So what Chelsea found is that, in fact, the advantages of accelerating reproduction are not specific to high adversity females. There was no evidence, statistical evidence for an interaction effect between early adversity and the timing and pace of reproduction. And that means that speeding up reproduction is helpful to fitness, but is not especially helpful under the experience of early life adversity. So, in answer to Chelsea's question, individuals who can anticipate dying young, at least in the baboons, do not seem to live fast or reap particular benefits from living fast. These results are counter to longstanding hypotheses in evolutionary medicine and psychology. And in some ways, maybe aren't so surprising. You know, in long-lived, slow-reproducing animals, including humans that invest in one offspring at a time, lifespan should really be the dominant driver of fitness and not the timing and pace of reproduction. All right, so one more story I want to tell you guys about sort of building off these developmental or behavioral results of early life adversity. And this is a project led by Stacy Rosenbaum, who is a postdoc in my lab last year with the considerable help of Shuxi Zheng, who's a PhD student in statistics at Duke, who developed new statistical methods to try to answer this question. And the question was, to what extent do social relationships help explain a link between the experience of early life adversity and adult stress responses? So unfortunately, it's pretty well known that the experience of early life adversity can alter stress responses across the life course, in early life, during adolescence, and also during adulthood. In this project, we focused in adulthood. And these dysregulated stress responses are often linked to chronically elevated glucocorticoid hormones, which in turn themselves have negative effects on health, such as immunosuppression or links to metabolic disorders or metabolic syndrome or cardiovascular disease. So while it's well known that early adversity can lead to dysregulated stress responses, the causes of those relationships are not well understood and are in fact subject to debate. So under one common hypothesis, the experience of early life adversity has strong and direct effects on adult stress responses. And in this case, early life adversity is the fundamental cause of this dysregulated stress responses in adulthood. So for instance, in this scenario, the experience of early life adversity may lead to differences in the development of the HPA axis, and that leads to differences in adult stress responses. In contrast, a social causation hypothesis proposes that the experience of early life adversity is not the direct cause of adult stress responses, and in fact, adult social conditions, in fact, this relationship is strongly mediated by adult social conditions. So this relationship, in particular, the idea here is that the experience of early life adversity makes it challenging for individuals to form strong and supportive social relationships, especially in adulthood. The lack of those strong and supportive social relationships then leads to dysregulated stress responses. So here the social conditions are the primary cause. So from many studies, we have evidence for sort of each piece of this arrow. We know that early life adversity can lead to social isolation. We know that social isolation or lack of social support can lead to dysregulated stress. But what we'd been missing was sort of the ability to put all of these together in exactly the same subjects in the same time and use a mediation statistical framework to disentangle the relative strength of what is passing through the effects passing through adult social conditions versus the direct effects of early life adversity. So we had the data to do this. We measured early adversity prospectively in 197 female baboons. We've already talked about the methods for doing that. And then we also looked at the strength of social bonds, and we looked at those annually across adulthood. We measured the strength of social bonds looking at female grooming relationships, which are a reliable predictor of social support. And then finally, we looked at adult stress responses using glucocorticoid hormone concentrations measured non-invasively in over 14,000 fecal samples. And this allowed us to measure basically trajectories of individual glucocorticoid hormones across adulthoods. It was the longitudinal data in social relationships and in glucocorticoids that required sort of new statistical methods that were developed by Xu Zheng. I'm not going to go into those methods now, but I will show you what we found. So first we asked, first what I'll show you is what is the total effect of early life adversity on glucocorticoids across adulthood? So regardless of what path it flows through, does it flow through social conditions or not? Just what is the total effect of early adversity on glucocorticoids? And what we found is that the experience of cumulative adversity leads to a 9 to 14% increase in glucocorticoids across adulthood. What I'm showing you here is an effect size plot, and I'll step you through it because I'm going to show you a few more. So on the x-axis here, we have the percent difference in fecal glucocorticoids. And then here on the y-axis, we have each individual source of early adversity, as well as two measures of cumulative adversity, the experience of no adversity versus one or more sources, or the experience of one source of adversity versus two or more sources. And you can see these are what is leading to sort of a 9 to 14% increase in glucocorticoids across adulthood. So we next asked, what is the relative importance of that direct path versus the path mediated by adult social conditions? Is there evidence for this social causation route? What we found is that, in fact, the direct effect of early adversity on adult glucocorticoids is 11 times stronger than the effect mediated by social relationships. So on this effect size plot, the green symbols are showing the effect size of the direct effect, and the pink symbols are showing the effect size of the mediation effect mediated by social relationships. The mediation effect was actually often significant, but it was very much smaller than that explained by the direct experience of early life adversity. So we next wondered, why was there such a big difference in the direct and indirect effects? And we have a couple of explanations for this. One explanation is that the effects of early adversity on the strength of adult social bonds were present, but were fairly modest. So in other words, this link in the path, the link between early adversity and adult social conditions was present, but small. So in this effect size plot, the x-axis is the difference in social bond strength. Out here, this dashed line shows us a 1 standard deviation difference. And so the experience of these sources of adversity is only leading to about 1/3 of a standard deviation change in this metric of social bond strength. Moreover, the link between adult social conditions and glucocorticoids, while statistically significant, was also relatively small. So here we can see the difference in adult glucocorticoids, again, is our effect size on the x-axis. And we can see that what this is showing us is the effects of a 1 standard deviation change in social bonds leads to about a 5% mitigation or reduction in glucocorticoids. But remember, we're not even getting up to a 1 standard deviation change in social bonds here. We're only getting about 1/3. So while these were significant, they were small. So together, what these show us is that at least in the Amboseli baboons, in this, posed in this way, early adversity does have large direct effects on glucocorticoid levels across adulthood, but these effects were not strongly mediated by social isolation in adulthood. And if we're going to try to translate this to human health, which may be a speculative thing to do, but let's do it anyway, I think that solving early life effects on health will likely depend on improving early life conditions, or that our results point towards this being one of the most promising strategies for trying to mitigate those relationships between the experience of early life adversity and dysregulated stress responses. So I know I'm preaching to the choir here, but hopefully what I've done is shown you that a long-term perspective on individuals across their lives can really shed light on some questions that have been burning a hole in ecological or evolutionary pockets for quite some time. And given the time, I should probably stop, but I would like to thank my collaborators, the members of my lab, and of course, my funding sources.
Great, thank you so much, Beth. There are a few questions in the chat that we'll start with on the microbiome stuff. First, Mark Haber asks to clarify what the pattern of intra and inter-individual similarity in the microbiome. So you touched on that it seems to be like seasonal differences, but could you talk a little bit more about that?
A little bit more about the, yeah, so I think, it's like what we're seeing is each, like the population is to some degree changing in concert across seasons. And you can definitely detect that signal in the data. But then if you go in and look at each individual's trajectory, the way each individual is doing it is slightly different. And it's not that they're not, they're not in totally unique microbiome space. They're all just sort of like in, what's the right way to explain this? They're sort of in, they're all in, like if you think of it as music, they're all sort of in different parts of the song at different times. So they're all in sort of they're all in shared community states. Well, that's not the right way to say it. There's sort of a global pattern of community states, and they're all sort of in each of these different states, but they're not all in them at the same time, if that makes any sense. Yeah, that was a bit stumbly.
All right, there's also somewhat of a follow-up or building on Mark's question from Jean Gilbert, who asked, could fluctuate, well, first of all, he says, what a data set, exclamation point. Could fluctuations in the microbiome—
I know, it's a crazy data set.
Yeah.
Yep.
Could fluctuations in the microbiome synchrony among individuals possibly be explained by fluctuations in diet niche partitioning? For example, in a good year with plenty of food, baboons might have larger niche partitioning or reduced competition, resulting in more dissimilar microbiomes where in bad years there's less food and no ability to partition.
Yeah, that is a great question that we would love to have more information on. We have information, so I have a few thoughts. So one is our experience as observers out there with the baboons, it does often seem that basically everyone's eating the same thing at the same time. Like they're all out there, everybody's eating corms or everybody's eating grass. There's sort of like one good thing, maybe not one, but a handful of good things at a given site. And because the group is all together, that seems to lead to a lot of synchrony in the diet. Our diet data are a little bit sparse, and so that we have a difficulty digging into fine-grained differences in diet. But we would love to use either more fine-grained behavioral observations, or we're working on metagenomic methods to look at diet composition in fecal samples. And that may shed a little bit more light on individual differences in diet, but it's certainly possible that that is explaining some of the idiosyncrasy, and we just don't have the data yet to detect it. Yeah, it's a great question.
I think Matthew has a question or two, so I'll let him to ask.
Yeah, well, they're kind of related questions. One is kind of a ask you to speculate kind of question, which is, in the next, let's say 15, 20 years, right? What kind of advances do you think that we're going to see in the study of microbiomes? And what do you think the prospect of connecting either causally or correlationally microbiome between individual differences in microbiome to fitness outcomes is? I think we're going to get there. And yeah.
Yes, I think, well, okay, so I have a few thoughts. So one is, I think the in terms of the future of microbiome work, we need to move away from the 16S stuff that I just presented, unfortunately. But so basically, we need to, and we already are moving to the ability to look at whole genomes. More challenging to do that of individual cells, but at least looking at strain level whole genome patterns and actually looking at the evolution of strains over time within and between individuals. That is like what I really want to do. Part of that will involve culturing. So it's funny, we had all these culture-free methods that liberated us to see a whole bunch of stuff, and now everyone's returning to try to culture some stuff, because that's actually the real way you can really look at the organisms and see what they're actually doing. So that's one thing. I also do have a lot of hope that there's scope for connecting the microbiome to fitness outcomes. I think that the way to do that now, and one way I'd really like to do that is by joint modeling, sort of jointly modeling microbiome dynamics and connecting those to reproductive outcomes or survival. So I do think it's feasible.
Well, staying on that topic, I'm gonna ask you to step out on the speculation branch a little, just one more step, which is, so you said a few times that microbiomes are really dynamic, which reminds me of the way that people talk about hormone levels. And so I'm thinking about what's important for an animal in terms of surviving. Do you think it is the actual, when we measure things, is it the makeup of the microbial communities that are important, or is it their ability to change their communities in the appropriate way that you think really matters for these individuals?
That is a great question. I would guess it's a little bit of both. It's probably not an either or, but I do think that the link between microbiome dynamics, like how fast can you turn the ship to work on a new resource, is an underappreciated and probably very important aspect of microbiomes, especially as they are linked to host health. One other thing that's also worth mentioning too is that causally linking the microbiome to host health. I mean, there's actually many studies that show quite experimentally quite good causal links between the microbiome and host health. In our system, it's very hard to tell to what extent is the microbiome driving health versus, and this isn't going to be impossible to disentangle, but is it driving health? Or are we just detecting bellwethers of an individual's behavior and overall physiology that are then predicting fitness? But whether they're actually causally linked to fitness, that I don't know if we'll ever see. But yeah, good questions. Yep.
All right, I think we'll end it there. I'd like to thank Beth and Guill so much for the stimulating and thought-provoking talks, and remind everyone that we'll be back next week, same time, same place. And again, please fill out that survey on how this series has worked for you. All right, thank you all.
Great, thanks guys.